Hi, I am Sean Touhey.
And I am Matthew Durham. In this experiment, we explored the expression of Mol11 in what is commonly known as baker's yeast.
Mol11 is the gene responsible for the physical transportation of maltose in yeast during the process of fermentation. Yeast is often used for this process to make wine, beer, mead, and the like. However, when glucose is introduced, Mol11 expression declines because glucose is easier to break down than maltose as it is less complex. Our hypothesis was, when more complex sugars are introduced, Mol11 expression should theoretically be increased. Since maltose, glucose, galactose, and fructose are all monosaccharides, the samples with sucrose will show the most Mol11 expression because sucrose is a disaccharide and harder to break down than monosaccharides. But since glucose is the simplest sugar, the glucose samples will show the least Mol11 expression.
Our experiment consisted of 4 major steps. First, we had to culture and ferment our yeast cells. Then, we had to extract RNA from those yeast cells. After that, we had to synthesize our cDNA from the RNA. Finally, we had to analyze our cDNA with real-time PCR.
All of our samples were supplied with the 2% maltose necessary for fermentation and therefore Mol11 expression. 13 samples total were run, with the following concentration of each sugar. Our control was only 2% maltose. 2% maltose was also introduced into each of the following samples. Fructose 1, 2, and 3%, glucose 1, 2, and 3%, galactose 1, 2, and 3%, and sucrose 1, 2, and 3%.
Here are listed our materials. You can see that none of them are of any note.
It is very important to use sterile technique throughout the entirety of this experiment. You don't want unwanted specimens in your experiment.
In each of our conicals, we had to pipette 5 milliliters of YP media. Then, we had to take yeast culture plate and with a wire hook, pick up some of the yeast cells. Then, we had to mix the cells in the conical with the YP media using the wire hook. After 24 hours, we continued.
The next steps in the yeast culturing process involved putting all the materials needed for fermentation into a 250 milliliter Erlenmeyer flask. The table on the right of the screen shows everything that went in there and the volumes of each object. We pipetted 500 microliters of the yeast growth into each flask after we put all the necessary sugar in it. We let it ferment for at least 3 days while the flasks were on an orbital shaker at approximately 75 rotations per minute.
In order to extract RNA, we needed some more materials. They are listed here.
To extract RNA, we had to extract 1,000 microliters from each fermented sample and put it into an epi tube. After we centrifuged the epis at 2000 RCF, which is equivalent to 2000 Gs, which is not 2000 rotations per minute, for 5 minutes. We poured all the excess liquid off the top of the yeast pellet at the bottom of the epis, carefully using a pipetter to remove any excess liquid. Store those yeast pellets at negative 80 degrees Celsius if necessary.
Continuing, we made a conical of lysis buffer with mercaptoethanol, which is very highly toxic. We had to put 600 microliters of the lysis buffer in each of our epis. We vortexed each of the epis and its contents until the cells inside appear lysed. Then, we had to put 1 volume of 70% ethanol in each epi.
Do note that the yeast pellets are very fragile and sensitive, so you have to be really careful and move very quickly while the yeast cells are not stored at negative 80 degrees Celsius. The mercaptoethanol lysis solution should be made by someone that knows how to safely handle chemicals, like a teacher, as it is very, very highly toxic.
Continuing further, we put 700 microliters of each sample into its own spin column in the RNA extraction mini-kit. We had to spin in a centrifuge at 12,000 rotations per minute for 15 seconds until the entire sample was processed. Adding 700 microliters of the wash buffer, which was provided in the kit and spun, continued our process. In a new collection tube, we put 500 microliters of the wash buffer too, provided in the kit, and we spun them again at 12,000 rotations per minute for 15 seconds, 2 times.
The last set of steps in the RNA extraction process involved putting a new epi under the spin column as the collection tube, adding 100 microliters of RNase-free water to the spin cartridge and spinning that. After incubating it at room temperature for 1 minute, after centrifuging, store the samples at -80 degrees Celsius if necessary.
Listed here are the materials and equipment necessary for synthesizing cDNA.
To synthesize cDNA, you need to make a PCR master mix and run that in the PCR machine. Shown on the screen is the master mix table for the PCR reaction. Pipette 10 microliters of the master mix into 26 PCR tubes. Then you're going to pipette 10 microliters of each sample into 2 different tubes.
Seen here are the PCR cycles that we used. After they were run in a PCR machine, we put the samples into a single epi. We tested each sample with a nanodrop machine in order to test the DNA concentration in nanograms per microliter.
Our final major step in this experiment was real-time PCR. Here are the materials needed for this step. Programming the real-time PCR machine takes a lot of time and patience.
Here is the real-time PCR master mix that we used.
Because our real-time PCR machine is only capable of running 48 well plates, we had to do 2 different 48 well plates to get sufficient results. Here are the layouts for those plates.
These next 4 slides are t-test charts from an Excel spreadsheet. We ran these to ensure that the averages between 2 different changes aren't because of random sampling errors. The probability value is seen at the second from the bottom. Multiply that number by 100 and that is the confidence number. This number must not exceed 5% to ensure that we can be confident about our results not being because of random sampling errors.
This is the expression chart. Each sample is compared exponentially to the control. For example, with the 2% sucrose with a value of about 10.5. That means that it is a 10.5-fold increase over the expression of the control.
From the data we received, we concluded that our hypothesis was proved to be correct. Sucrose yielded the highest expression of the MAL11 gene, with an average 96.27% certainty that it was not based on a random sampling error. The glucose samples also yielded the lowest expression. This had an average 98.19% certainty that it was not based on chance. Therefore, we can accept our hypothesis and reject the null hypothesis.
However, we did find that only the 2% sucrose yielded very high expression. We do not know why it did not work as well for the 1 and 3% samples. However, the top 3 samples yielding the highest expression were in order from highest to third highest: sucrose 2%, sucrose 3%, and sucrose 1%.